My name is Sudarshan Pingle, and I lead a research lab affiliated with the Department of Genome Sciences at the University of Washington, the Broughton Beatty Institute, and the Seattle Hub for Synthetic Biology. The collaborators on this work were Jason Dore, J.B. Lalane, Riza Daza, and many others. So there are two questions about genome structure that really motivated the study. The first is to understand what fraction of the genome is actually functional. Are there hidden constraints or sequence elements or features that are really important for our genome to function that we don't know about? The second is to understand the impact of structural variation. Now, these are usually sequence alterations that are larger than 50 base pairs and include things like deletions, translocations, inversions, as well as novel sequence insertions. Increasingly, in the last decade or so, extrachromosomal DNA circles are another form of structural variation that have been implicated in various forms of cancer and disease progression. However, understanding the impact of structural variation has been challenging, and this is for two reasons. One is that they have about 100-fold lower rate of occurrence than single nucleotide variants in human populations. Secondly, if we wanted to take another approach, which we often take in genetics, which is to engineer these variants into cellular models to understand their impact, it has been very, very difficult, owing to the nature and size of these structural variants, to engineer them into these models, even with tools like CRISPR. And so this is really what we wanted to solve. We wanted to understand and ask if we could engineer and characterize mammalian structural variants in one single experiment. So as an inspiration, there are these systems called site-specific recombinases that can induce rearrangements between DNA sequences. The most famous example is Cre-Lox, where a protein called Cre, a recombinase, recognizes two 34 base pair sequences called LOX-P sites and can rearrange the sequence between those two sites. So the nature of the rearrangement that results from the action of Cre recombinase is determined by the orientation of the two sites. And this feature had actually been used previously in the synthetic yeast project to develop a method called scramble. You can see that starting from one parental configuration of these segments in the genome, just by turning on the Cre recombinase, you can get much smaller as well as much larger and inverted rearranged sequences from that one parental allele. However, adopting this to mammalian systems is challenging for two reasons. One is, how do we introduce these many LOX-P sites into the genome? De novo synthesis of whole mammalian chromosomes or even genomes is just impractical at this current moment, owing to cost and the size. Secondly, in all instances of inducing these types of structural variants, people were characterizing the impact using whole genome sequencing by isolating clones. Now, while this works really well for systems with small genomes like yeast with a 12 megabase genome, but if you wanted to do this in the scale of mammalian genomes and your readout relied on whole genome sequencing after clonal isolation every time, this dramatically limits the scale. And so we wanted to solve this problem. So the way we went about this was coming up with this method called Genome-Shuffle-seq that allows multiplex generation of mammalian structural variants, as well as their characterization in one experiment using single-cell sequencing. So how does this work? The core of the technology is the design of this thing that we're calling the shuffle cassette, which contains 4 features. One is a site-specific recombination site. This could be from Cre-Lox or other types of recombinases like BXB1. Immediately flanking the recombinase site are 2 random barcodes. Flanking those are phage polymerase promoters like T7 promoter that are completely inactive in mammalian cells, but then you could turn on the expression of those promoters using either in vitro transcription with that polymerase or in situ transcription on fixed cells. These are cloned into a transposon vector in this initial experiment, and we're cloning a very, very complex library such that there's a huge pool of these recombinase sites, each associated with random barcodes that serve as somewhat of a unique marker of each recombinase site. We then introduce this library of shuffle cassettes into our target mammalian cells using transposition at high MOI, or multiplicity of infection, which means there's many, many integration events of these shuffle cassettes in any individual cell. And in each cell, there's a unique combination of barcodes plus insertion sites. So for example, let's say in one particular cell in this population, we had these 4 insertions at these locations. You could do in vitro transcription on genomic DNA extracted from this population. And if you sequence those short RNA transcripts that come out of these T7 promoters, what you'd be able to do is associate the barcodes that are present with the neighboring genomic sequence. For example, here you're saying that barcode 1 and barcode 2 is found at this location, barcode 3 and 4 here, and so on. Now, once you've made this giant table of where barcodes are found in your population, all you have to do then is turn on your recombinase. And if you want, throw the cells through differentiation or different selection conditions. And then we can assay what structural variants are induced either in bulk or single cell. So let's say in this population, we induced this particular rearrangement that came from an inversion and deletion. Because site-specific recombinases are site-specific, it leads to a change in the set of barcodes that are found together on the same molecule. So for instance, you'll see that in the parental population, barcode 1 and barcode 2 were found on the same molecule. But in this rearranged population, barcode 1 and barcode 3 are found in the same molecule. So by sequencing what barcodes are present in the rearranged population and comparing it to our original parental population, we could actually infer the nature of the rearrangements that we're inducing. Now, these can just be read out simply by PCRing up these shuffle cassettes and doing bulk amplicon sequencing. Or if we do in situ transcription with T7 polymerase on fixed cells, we again reawaken these short transcripts, which basically capture a RNA fingerprint of the genotype of that cell. And so we can capture these short RNA molecules along with the rest of the transcriptome in your favorite single cell sequencing assay. Now, again, just to emphasize, this does not rely on any whole genome sequencing and allows us, in theory, to generate thousands of structural variants and read them out very, very simply in bulk or in single cells. So just to show that this scheme works, we initially did this experiment in mouse embryonic stem cells. And this is one particular example of a mouse embryonic stem cell population that has two different alleles, a black 6 allele and a cast allele, which have one variant every 125 base pairs or so. And therefore, we can actually get allele-specific resolution of these integration sites. So you can see along chromosome 1, we have a whole bunch of different variants. And you can very neatly see that we can pick apart the cast insertion based on the variants that are found in the genome, as opposed to one that's on the black 6 allele. In our initial experiments, we were able to map the integration sites of about 5,000 or so, and about 80% of these could be assigned to one of the two alleles. Now, here comes the money experiment, which is we transfect in this population that contains LOXP sites that are targeted by the Cre recombinase, either with the targeting recombinase Cre or the non-targeting recombinase BXB1 as a negative control. We let the cells grow out for 3 days and then simply just PCR up these shuffle cassettes to see if we see new barcode combinations. And what was really encouraging was that in 3 different concentrations or amounts of Cre that we were transfecting into these cells, we were able to see thousands of new barcode combinations, each reflecting a rearrangement that was induced in that population, whereas we saw very few of these new barcode combinations in the negative control condition, which told us that we had really great signal to noise, and we were able to both induce and map thousands of rearrangements in one population in one experiment. These are of all kinds. In the subset I'm just showing you here, we have deletions, inversions, as well as translocations, and these involve all the chromosomes that are found. However, this is where we ran into a bit of a roadblock, which is we discovered that Cre-mediated structural variants at symmetric LOXP sites really did not persist. They seemed to induce a whole amount of toxicity to cells, and we were never able to isolate cells that were long-lived but had induced structural variants in them. However, we discovered that switching to a different recombinase called BXB1 and using its asymmetric sites were actually way more tolerated, and we were able to recover cells that had structural variants over longer periods of time. So what are the types of selection events that we see? So one, we found that deletions spanning centromeres were massively selected against. So here just showing you at day 3, day 6, and in bottleneck populations, we expect deletions and inversions to be induced at roughly even frequencies, which is what we see at the early time points. But at this late time point, we basically don't detect very much, if at all, any reads that support the deletion of a centromere, which I think kind of makes sense, and showing that using this method, we can actually pick up real biological signal. Secondly, and this is quite intriguing, depending on the relative orientation between the different sites and their sites of integration relative to the centromere, we can induce either balanced or unbalanced translocations, which can lead to acentric or dicentric chromosomes. In the case of balanced translocations, there's actually no loss of genetic information whatsoever. And what we see is both in K562s, which are the human cancer cell line, as well as in mouse embryonic stem cells, we see that both balanced and unbalanced translocations are induced at roughly even frequencies, again, at these early time points. But over time, the unbalanced translocations are actually much more depleted. And specifically, the acentric form of unbalanced translocations are more depleted over time than that dicentric version. Lastly, we actually see that if you have a deletion that occurs between true recombinase sites, you also pop out, in addition to the deletion in the genome, an extra chromosomal DNA circle. And what was really cool was that we were able to see hundreds of these extra chromosomal DNA species that we were able to launch in an unbiased fashion across the genome. In early time points, we were able to see both the ecDNA as well as the genomic copy, but over time, we lose the ecDNA, which is kind of what you'd expect for a genetic element that doesn't have the mechanisms to segregate through cell divisions. So these data basically show that, hey, look, we can induce these structural variants in a multiplex fashion in one experiment and read out the impact of these structural variants in one simple bulk assay. But what we also really wanted to do was ask if we could do single cell analysis of the induced structural variants, and our assay is actually set up to do that. So let's say you had an integrated shuffle cassette in the genome. If you fixed your cells and then did T7 transcription while the cells were present, you would capture first the endogenous mRNAs, but then you'd also induce these short transcripts that are coming from the shuffle cassettes. And remember, the identity of these barcodes actually reflect whether there was a rearrangement or not. Now, if you can put it through 10x Genomics feature barcoding, these beads have poly-DT primers that allow you to capture the endogenous mRNAs, but they also have oligos with these capture sequences, CS1 and CS2, that allow us to capture these T7 transcripts. In one experiment, we were able to find 53 different clones based on the complement of T7 barcodes that were found within them. We realized that within this clone, there was a 500 kb deletion to one copy of chromosome 15, and that contained these genes, out of which 3 were actually expressed in these K562 cells. Now remember, K562 cell is triploid and chromosome 15 is triploid in these cells. And if you delete one copy of triploid chromosome, you expect a 33% reduction in that particular sequence. And in the single cell data, what we actually see is if you compute the fold change between cells that contain this deletion versus not, and look for 3 gene windows and what the mean fold change is, you find that there's only one outlier, and this is actually the 3 gene window that encompasses the deletion. And the fold change is exactly what you'd expect, which is 33% reduction from the wild type. So this actually shows us that we can both very specifically detect structural variants in a single cell, but also assess the impact on gene expression. One major challenge so far is that the proportion of cells that contain structural variants is still quite low. Even in our best experiments, it's about 4 or 5% of cells, which means we're throwing away a lot of sequencing just on wild type cells that are not very, very interesting. And so this is a major focus for us right now. One way that we think we can do this is by including selectable marker cassettes in our designs that basically reconstitute selectable markers, either an antibiotic resistance or a fluorescent marker that's specific to when the rearrangement is actually induced. What we're hoping is to do then perturb-seq like screens for structural variant impact. And what we're hoping to do then is large screens of structural variant impact in a variety of different contexts. We're also very interested in expanding the kinds of molecular features that we can profile in addition to RNA. We're working on DNA accessibility as well as chromatin conformation capture. Finally, I want to say that maybe this is the start of what I like to think of as a minimal mammalian genome project. So we know that each one of our cells carries 6 billion base pairs of DNA, and I would argue that something less than 6 billion bases is actually required for every cell to function. And we'd like to understand how much, because minimal genomes, I think, have exciting implications both for fundamental biological discovery, but also for biotechnology in cell and gene therapies, as well as bioproduction. I would be remiss if I didn't point out that this paper came out with a paper from Leo Parts, George Church, Jonas Kopel and Rafael Ferreira, and these two studies really stand together to show that whole genome shuffling in mammalian genomes is possible.